My name is Abdul Bar. I'm a PhD candidate at the University of Adelaide and also a lecturer at the University of Northern Technical University in Mosul, Iraq. I'm going to talk today about rapid screening method for identification of bovine milk mycoplasmas and Acholeplasma laidlawii. To start with a brief introduction, Mycoplasma mastitis is increasingly generating considerable interest in bovine dairy industry. Mycoplasmas are the smallest bacteria yet discovered, and it is related to class molecules. In this class, there are several genera that can be of significant importance in human and animal health, such as Mycoplasma, Ureaplasma, and Acholeplasma. There are more than 200 species of Mycoplasma yet discovered. Some of them are saprophytic mycoplasmas and others are pathogenic. And of these pathogenic mycoplasma, there are several species that cattle-associated, like in Bovis, Californicum, Bovigenitalium, Alcalensis, Boviculi, Mycoides subspecies Mycoides, Dispar, Acholeplasma, and Ureaplasma diversum. It is common knowledge that Mycoplasma can cause clinical mastitis when there are obvious clinical signs on the udder or the animal, and it can be acute, subacute, or chronic, and subclinical mastitis when there are any clinical manifestation, but the changes just in the milk composition, mainly somatic cell counts and milk yield. Mycoplasma, in my cases, tends to cause subclinical rather than clinical mastitis. It is often thought that the mechanism of milk alteration is attributed to the pathogen severity, proportion of involved mammary glands alveoli, interference with blood and hormonal nourishment to these alveoli, and epithelial integrity disruption and milk decomposition due to enzymatic activity. These bacteria have the tendency to cause mastitis either as an individual or co-invader pathogens. The economic consequences of Mycoplasma infection in cattle are enormous. They include the cost of loss of beef weight, the cost of culling of diseased cattle, significant decreases in milk production, the cost of implementation of control procedures, as well as the cost of diagnosis and treatment. In the United States, for instance, the cost of Bovis-associated diseases is more than $100 million US dollar each year, and more than this number is estimated in Europe. In May this year, the New Zealand government decided that it would attempt to eradicate Mycoplasma bovis diseases at an estimated cost of 886 million New Zealand dollar. Mycoplasma species are fastidious, slow-growing organisms, and it can take up to 15 days to obtain the culture results. In addition to that, the special culture requirements to kill and inhibit all the gram-positive and gram-negative bacteria. As a result, Mycoplasma is usually a missing component from routinely mastitis screening. And finally, the difficulties in survivability of these bacteria should be also considered. The study aimed to develop a rapid screening and accurate screening method for identification of Mycoplasma mastitis, investigate the relative merits of conventional culture-based method versus DNA amplification for detecting Mycoplasma mastitis in bovine milk, and finally, to discriminate between field isolates of different species of Mycoplasma, field isolates of Mycoplasma and Acholeplasma laidlawii, using high-resolution melting curve assay, or HRM. For that, a total of 368 milk samples were collected at individual cow level from a single commercial dairy farm in South Australia, particularly in Mount Gambier. This is the state of South Australia, and this is Mount Gambier, beautiful city located closely to Victoria State. We collected those samples at cow levels, as I said, and samples were kept on ice and sent immediately to the University of Adelaide in Adelaide. This farm had a history of high somatic cell counts and multiple antimicrobial treatment failure. In the first experiment, we developed universal primers targeting 16S ribosomal RNA gene of all possible mastitis-associated molecules at genus level. DNA was extracted directly from milk to save time and money. Then we compared the results with the culture-based method and also with a previously published PCR-based method. We used ATCC strains of some molecules as positive control, and we also confirmed all these results through Sanger sequencing. These are some of the primers that we used in this study. Of 368 milk samples collected at individual cow level in single dairy farm in South Australia, 52% tested positive for culture-based method. Our universal designed PCR yielded 73% in comparison with 70% using previously published PCR assay. So our universal PCR demonstrated highest or the best concordance with Cohen kappa, like good Cohen kappa, with the previously published species-specific PCR. The detected species were Acholeplasma laidlawii, M. bovis, M. bovirinus, M. arginini, and M. bovirinus, and uncultured mycoplasma as well. Infection by 2 or more of these above-mentioned molecules showed the highest prevalence. In the second experiment, we compared the effect of 6 different groups according to their culture and PCR results with the healthy cows on milk composition, including individual M. bovis, M. bovirinus, Acholeplasma laidlawii, co-infection molecules, 2 or more of these mycoplasmas, and other mastitis, major mastitis pathogen, including strep, staph, E. coli, and coliform. All these, we put them in one group and compare the effect on the milk composition with other groups, and of course the healthy cow group. A database of individual cow yield production parameters, including somatic cell counts, volume, protein percentage, total milk solids, and fat percentage, were analyzed per each group. Results have shown that significant difference in test day somatic cell counts was detected between almost all groups except the group of Acholeplasma laidlawii and healthy cows group. Correspondingly, test day milk yield was similarly affected significantly with co-infection molecules, other mastitis pathogen, and mixed infection between those groups. Also, total milk solids decreased significantly in similar groups in comparison with the healthy cow group. Fat percentage was significantly decreased just in co-infection molecules group. However, no differences were observed in all other tested groups. Protein percentage increased significantly in molecules co-infection group and mixed group, while other mycoplasma groups showed no protein differences. For more explanation about these results and the discussion of these results, we published a paper in BMC Veterinary Research, and it is available on their website. In the third experiment, HRM profiles were developed using Ecosystem by Illumina. The software characterizes DNA samples according to their dissociation behavior as they transition from double-stranded DNA to single-stranded DNA with increasing temperature. Here, we use the same primers that we used in the first experiment, and we selected 5 representative samples per each group were selected for this study. Results showed that the real-time PCR-HRM assay was able to detect and discriminate between different field isolates of molecules. This assay showed clearly distinct melting curve profiles of Acholeplasma laidlawii, which is in purple color, Mycoplasma arginini, this one with a green color, and Mycoplasma bovinis, the blue one, and M. bovis, the yellow in color, the first one, and uncultured Mycoplasma. So for these, as I said, we used ATCC as a positive control in addition to sequencing. This software can also generate, as you can see, differences melting curve, and it can also generate digital values that makes the comparison easier. In conclusion, due to rapidity and higher sensitivity compared to the convention culture method for surveying Mycoplasma mastitis in dairy herds, we recommend screening of milk samples using our universal PCR method. The real-time PCR-HRM assay was able to identify and discriminate between mastitis-causing mycoplasmas and other molecules, which are often considered as milk contaminants. Results of our study indicates that co-infection Mycoplasma mastitis cause similar effect on milk composition due to other conventional mastitis pathogens. Finally, I would thank the School of Animal and Veterinary Sciences, my supervisors, Dr. Kira Petrovsky, Farhid Hamadzada, and other authors, HCED in Iraq, the sponsor organization, farms owners and the staff, and also Southeast Vets for their contribution in my study. And thank you so much for your listening.